Now we shall talk about the DNA replication synthesis of the leading strand. Story so far is that when an appropriate signal reached the cell, it prepared itself for DNA replication, for entering the cell cycle. There was an arrest in the G1 phase. Then the cyclin-dependent kinases, retinoblastoma protein, all of them interact and push the cell from the G1 phase to the S phase. In the S phase, the initiation of replication starts at ORI, which is AT rich. The helicase unwinds the DNA to form the replication bubble. The replication bubble can be few thousands of base pairs long. The replication bubble moves in both the sides. Single strand binding proteins bind to the DNA to prevent premature annealing. The primase, along with the helicase, lays one primer for the leading strand and multiple primers for the synthesis of the lagging strand. Now let's understand again the DNA polarity. The DNA polymerase requires a primer to provide the 3-OH group. So this end is the 3-OH group. This end has the 5-OH group. This is the 5' end. This is the 3' end. Every base pair that gets attached attaches via the 5' end and provides a 3' OH for the next nucleotide to attach. So the strand which is being synthesized is going to be synthesized from the 5' end to the 3' end, which makes the template DNA 3' to 5' orientation. The DNA polymerase is the main player which is involved in the DNA replication. In the prokaryotes, there is DNA polymerase 1, 2, and 3. In eukaryotes, it's a bit more complex. So we have the alpha, beta, gamma, delta, and epsilon. In prokaryotes, the main enzyme is DNA polymerase 3. DNA polymerase 2 edits and does the proofreading of the DNA. What's the meaning of proofreading? Proofreading means check up whether the correct base pairs have been added, and if not added, it's supposed to remove it. DNA polymerase 1 is responsible for repair of any DNA damage. In the eukaryotes, on the other hand, the delta uh, polymerase synthesizes the leading strand. The alpha synthesizes the lagging strand. The proofreading, which was in DNA polymerase 2 of the prokaryotes, is with the beta. The gamma is involved in the mitochondrial DNA replication, and the epsilon is involved in the DNA repair. So you can draw an association between the prokaryotic polymerase and the eukaryote polymerase. The mechanism is simple. The DNA polymerase binds to the strand, to the template DNA. The pCNA clamps the DNA to the DNA polymerase. The DNA polymerase keeps synthesizing the DNA from the 5' to the 3' orientation in a continuous stretch to produce the leading strand. There is a difference in the production of the lagging strand, which we'll cover it up in the next video.